Students, welcome back to a new video of Sunanda's tutorial. In this video we will discuss about the life cycle of Penicillium. So Penicillium, at first the mycelium or morphology of Penicillium. The mycelium or the body of the Penicillium is well developed, copiously branched. It is composed of colorless, slender, tubular, branched and septate hyphae. The hyphae of Penicillium have septa or cross walls between them. The hyphae run in all directions on the substratum and become intertwined with one another to form a loose network of hyphae constituting mycelium. Some of the hyphae may even grow into interior of the substratum and the rest spread on the surface. So the individual branches are called hyphae and all togetherly called as mycelium. The whole body is called as mycelium. The former secrete enzymes and absorb food material. The hyphae that grows inside the body of the substratum absorb nutrition from their body. These are called hostorial hyphae. Hostorial hyphae. Okay. Next, the aerial hyphae receive nourishment through hostorial hyphae and produce reproductive structure. So the hostorial hyphae helps in the absorption of nutrition and aerial hyphae helps in the reproduction. Becker in 1944 observed anastomosing between hyphae of two mycelia resulting in heterokaryotic mycelium. So sometimes two hyphae fuse with each other to form heterokaryotic mycelium. The mycelium in a few species may develop into sclerosium. Okay. The hyphae constituting mycelium are septate and cells are short. The septa between cells have central pore between them. That means when they have cross walls, these walls have central pore that connects two cells. Okay. Next, through the pores protoplasm flows from one cell to the another cell. Next, so it is a portion of hyphae enlarged to show the detailed structure of the Penicillium hyphae, a portion of hyphae. Next, reproduction. Penicillium reproduces asexually as well as sexually. The asexual stage, however, is dominant. That means during favorable condition, Penicillium shows asexual type of reproduction and constitute usual mode of reproduction, and sexual stage is very rare. Asexual reproduction mainly by vegetative method, mostly by fragmentation, and secondly by spore formation or sporulation. There are generally two types of spores are formed by the Penicillium species. One is conidiospore and second one is oidiospore. So let's discuss. At first, vegetative reproduction, I have already told you, the vegetative reproduction is the most common method of reproduction and it is the easiest one, and by the fragmentation. The hyphae break into several segments. Each segment develops as a new organism. So it is basically the basis of vegetative reproduction by fragmentation. Next, sporulation. Normally it takes place by the formation of a non-motile asexual spore, the conidia, which are produced exogenously. That means outside the body of the Penicillium. The asexual spore, conidia, are produced exogenously. That means outside the body of Penicillium. Next, Penicillium multiplies repeatedly by this method during the growing season. Okay. Next, conidiophores. The conidia or conidiospores are produced by a very specialized organ called as conidiophores. Next, in in the species with branched conidiophores, the ultimate branches which bears the tops of flask-shaped sterigmata or the phallites are called as medullae. The lower branches which supports the medullae and form a part of penicillium are called rami. The conidia are formed within narrow tips of flask-shaped phallites. Conidium initials is formed by distension of tubular tip of phallite. The phallite nucleus undergoes repeated mitosis. Okay. So these are the phallites, and by repeated mitosis, the conidia develops. Okay. Next, phallites form the conidia through mitotic division, creating chains of conidia and that are dispersed by the wind. And when they fall on a suitable substrate, the conidia germinate and forming a germ tube eventually develops into a new mycelium or new fungus, new penicillium. So this is how they reproduce asexually by conidiospores. Some scientists also observed oidia formation in penicillium. So the mycelial hyphae of penicillium, when made to grow, immersed in a sugary solution, they divide by additional septa into short uninucleate segment. Sometimes they develop septa in their cells and the segments become rounded and separate as thin walled spore like structure, and those spores are called as oidiospores. And it is the second type of spore which is produced by penicillium and observed by some scientists. Next, sexual reproduction. The male and female organs are called as antheridia and ascogonia respectively. The male sex organ is called as antheridia and female sex organ is called as ascogonia. Ascogonium at first, the female sex organ. It is long, erect and multinucleate, unseptate, aseptate and tubular structure. At its upper end, it may be curved like the handle of an umbrella. It arises as lateral outgrowth from any cell of vegetative mycelium. It is the female sex organ or ascogonium. Next, antheridium. Antheridium is the male sex organ. It is slender, uninucleate, hyphal branch originates either from adjacent cell of the same hypha which gives rise to ascogonium, that means near ascogonium, or from a separate neighboring hyphae. It is the antheridial hypha. The antheridium born on the antheridial branch. It grows off coiling loosely around ascogonium, making several turns about it. The distal end of male branch becomes slightly inflated and eventually cuts off as an antheridium by the septum. The antheridium is short, terminal, club-shaped and uninucleate structure. Okay. Next, plasmogamy. So after the coiling of antheridium around the ascogonium, its union takes place, and it is the union of two protoplasts which brings the compatible nuclei close together in the same cell. The tip of the antheridium comes in contact with ascogonium, and the point of contact, the double wall, both the wall of antheridium as well as ascogonium, they dissolves. Through the common pore, two protoplasts come in contact. The female nuclei in the ascogonium, however, arrange themselves in pair. Sometimes the female nuclei, two female nuclei close with each other. The pairing of the female nuclei in the ascogonium is called autogamy. So when male and female nuclei come together, they are called as plasmogamy. But sometimes two female nuclei come together called as autogamy. Each pair is called, however, both of the pair is called dikaryon. With the establishment of dikaryon, haplophase ends. Haplophase of this Penicillium life cycle ends and the dikaryophase starts in the life cycle. Next one is post-plasmogamy. So after plasmogamy or autogamy, it follows by the septation of ascogonium. So what is the septation? Each segment has pair of nuclei. Whether it is autogamy or plasmogamy, they both results in a pair of nuclei. Meanwhile, entangled sterile hyphae grows off around the sexual apparatus. Some sterile hyphae grows around them and form a total apparatus-like structure and form an investment of loosely interwoven hyphae and the sexual apparatus and afford protection to the structures developing within to provide protection. Next, so this is the whole structure. Young ascogonium, female branch, the male initial surrounds the ascogonium. Then it is the mature ascogonium, antheridium surrounding the mature ascogonium. Then these cells are all dicaryons. All the cells have two nuclei, whether after plasmogamy or autogamy. Next, development of ascogenous hyphae. So as a result of stimulus or formation of plasmogamy or autogamy, one or more lateral outgrowths arise from some of the binucleate segments situated in the middle of the septate ascogonium. Each outgrowth is called ascogenous initial, and these are binucleate. Obviously after plasmogamy or autogamy they are binucleate. Each ascogenous initial develops into branched ascogenous hyphae composed of binucleate cells. Next, formation of asci. Asci in many species are developed directly from those binucleate cells towards the tip of the branches of ascogenous hyphae, and consequently they are arranged in short chains. But in some species, this ascos formation is direct. That means binucleate cells are directly developed into ascos mother cell and asci. No crozier formation is there. The binucleate cell enlarges into sac-like structure called ascos mother cell. But in some cells, in some species, terminal binucleate cells of the ascogenous hyphae curl over to form a hook-like structure which is called as crozier. The binucleate cell in both the cases during direct and indirect formation enlarges to function as ascos mother cell, and this ascos mother cell is the last structure of dikaryophase. Two nuclei in it eventually fuse, and this is called as karyogamy. With karyogamy, the dikaryophase of life cycle ends and the diplophase begins, because now it is 2n, because n and n fuse with each other by karyogamy. The septate ascogonum, ascogenous hyphae fusion or diploid nucleus is the young ascos. It is the only diploid structure in the life cycle and represents the short-lived transitory diplophase. Next, differentiation of ascospore. Now the young ascos enlarges. Its diploid nucleus undergoes 3 successive divisions. The first and second divisions are meiosis. The third one is mitosis. The resultant 8 nuclei are all haploid in nature. Okay. Meiosis have 2 phases, meiosis 1 and meiosis 2, which is followed by mitosis. So all togetherly it is 3 divisions, 2 meiosis, 1 mitosis, resulting in 8 nuclei, haploid nuclei. So this is the diagrammatic representation of ascos or ascospore development. So these are the ascospore. Then formation of ascocarp with the septation of ascogonium, development of ascogenous hyphae. A large number of sterile hyphae grow around sexual apparatus. The encithing sterile hyphae get interwoven to form a hollow ball-like structure. The peridium, which surrounds and protects the ascogenous hyphae as they grow and branch within, this is called ascocarp. This whole structure is called as ascocarp, and this is the fruiting body of this penicillium. The peridium or sheath is thicker than, it is the peridium thicker than the ascocarp of Aspergillus, consists of loosely interwoven hyphae. This entire structure is spherical and have no opening. Such closed ascocarp is called as cleistothecium. It is very, very important. The fruiting body or ascocarp with no opening is called as cleistothecium. Closed ascocarp is called cleistothecium. Next is discharge of ascospores. This whole fruiting body contains the ascospores, which are haploid in nature, and these are the structure from which the new generation will going to develop. At maturity, the wall of asci dissolves, the ascospore released. The liberated ascospores, each shaped like pulley wheel, float in nourishing fluids formed by degeneration of inner wall of peridium, the walls of asci and ascogenous hyphae. Okay, so these ascospores are actually float inside the fruiting body, and this fluid or nourishing fluid formed by degeneration of inner layer of peridium, walls of asci and ascogenous hyphae. Now ascospores absorb nutrition and mature. The mature ascospores are finally released by the decay of other wall of peridium and released into the environment. Then they fall on, after falling on a suitable substratum, they germinate, forming a germ tube, and finally mature into a new organism. Okay, so it is the diagrammatic representation of the life cycle of Penicillium. So Penicillium mycelium, which is haploid, so as they develop ascogonium, which is the male sex organ, and antheridium, the female sex organ, after plasmogamy. So male and female nuclei after pairing forms the dikaryophase, and after the formation of dikaryon, they fuse with each other by karyogamy, and karyogamy, after karyogamy the diplophase starts, that means 2n, which is very short lived and represented by the young ascus and ascocarp. From inside the ascus, ascocarps are formed, and ascocarps, the ascus mother cell undergoes meiosis, and after meiosis ascospores are formed, and ascospores after maturation released into the environment, and after germination they form a new Penicillium mycelium. It is all about sexual reproduction, but by asexual reproduction by means of spores or vegetative reproduction. They reproduce vegetatively by fragmentation, or asexually by cnidiospores or oidiospores. So it is all about the life cycle of Penicillium. I hope you will get benefit from this video, and if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.